disruption of RLA metabolism has become a central disease mechanism since the identification of TDP43 and RNA binding protein as a major protein that is aggregated in both ALS and FTD. So then in the second part, I will discuss our recent efforts to determine the contribution of STAT-MIN2 in disease. And we have recently shown that STAT-MIN2 is an important RNA target of TDP43 and is altered in patients with TDP43 proteinopathies. And lastly, I will discuss our efforts to model different disease mechanisms that have been proposed in C9orf72 ALS FTD. And so C9orf72 expansion is the most common genetic forms of both ALS and FTD. And we have used different mouse models and cellular models to test different mechanisms and to develop therapeutic strategies. So ALS is a neurodegenerative condition characterized by progressive paralysis. And this is really due to the loss of the upper motor neurons and lower motor neurons that do innervate from the cortex to spinal cord and to the muscles. And the loss of these motor neurons lead to paralysis that is fatal, usually within a few years. So in US, it's called also Lou Gehrig's disease from the famous baseball player who first raised awareness about this condition. In France, we call it Charcot disease from the neurologist who first described the condition more than a century ago. It's clearly clinically different from okay, this screen is stuck. So it's clinically different from frontotemporal dementia that is characterized by language and behavioral dysfunction. And for a very long time, as I said, we thought that these two conditions were distinct, and the genetic causes actually that were known. So for whatever reason, my slides are not moving. So 1 second. Okay, I'm going to try like that. You tell me. So as I said, the genetic causes were really different. So in 1993, a gene called SOD1 was identified to be cause of ALS, and mutations in tau and progranulin were known to be familial causes of frontotemporal dementia. But actually, over the last decade or 15 years, it's been clearly recognized that there is an overlap between these conditions. And in particular, some patients can have both ALS and FTD at the same time. And in some families, you can see individuals with either ALS or FTD. And the advance in our understanding of the genetics of this disease clearly show that many genes can be mutated and give rise actually either to ALS or to frontotemporal dementia in a mechanism that we actually don't really understand what would lead to one or the other disease. And I've already mentioned, so this there's a large expansion in the C9orf72 gene that really represents the most common genetic form for both ALS and frontotemporal dementia. However, our recognition that these conditions are overlapping also came from post-mortem tissue analysis and pathological studies, where we knew for a long time that both ALS and FTD patients accumulated ubiquitin-positive inclusions. And there was this breakthrough discovery in 2006 that a major protein in these ubiquitin-positive inclusions is actually TDP43. And so TDP43 is normally mostly nuclear, and you can see in affected neurons, it's mislocalized to the cytoplasm where it does aggregate. And that was a major discovery because, as you can see in ALS patients, almost all patients, including sporadic cases, do have TDP43 mislocalization, and actually a large proportion of patients with FTD also do have TDP43 pathology. And the direct role of TDP43 in disease was also confirmed by the identification of rare familial causes of ALS and FTD with mutation directly in the gene encoding TDP43. So when these patients have mutation, basically the pathology looks similar with this abnormal mislocalization. And so very early on, different disease mechanisms were proposed based on the observation in patient tissues. And you see, normally it's TDP43 is mostly nuclear, but in some neurons, you can see this very specific nuclear clearance that clearly suggests a loss of nuclear function of TDP43. But there are also these cytoplasmic dense aggregations that could confer a toxic gain of function. And better understanding the contribution of these mechanisms is very important for therapeutic development that would aim either to restore TDP function or to prevent mislocalization and aggregation. And major efforts in the field are really trying to target these different mechanisms. So I mentioned this is an RNA binding protein, and when it was identified to be altered in ALS and FTD, it was already known that this protein was involved in several steps of RNA processing, as are maybe many RNA binding proteins. And so it's mostly nuclear, where it has a role in splicing and transcriptional regulation, but it does normally shuttle to the cytoplasm and was shown to be included in stress granule and also to play a role in RNA transport and local translation. However, when it was found in 2006 and in the years following that, really only a handful of the RNA targets of TDP43 were known. And so a very important question was which RNAs are bound and regulated by TDP43. And this is what the question that we addressed, as well as many other labs in the field. In particular, for me, it was during my postdoctoral fellowship with Don Cleveland and collaboration with Magdalene Poliminido. We used different genomic approaches to identify TDP43 binding sites. So we use ClipSeq, and we use also RNA-seq and microarrays to determine what are the consequences of losing TDP43, in particular in the mouse brain, with alterations in expression and splicing of many RNAs. And from that, it was very clear that TDP43 does not bind only a handful of RNAs, but actually binds thousands of them. And that losing TDP43 has major consequences in RNA processing. And I think now the field, from these different efforts in many different models, the field has a good set of RNAs that we know are dependent on the function of TDP43. And here is an example, sortilin-1, that when you knock down TDP43 in mouse brain, you include more of a specific exon, while the transgenic mice overexpressing TDP43 include less of this exon. I'm taking this example because independently, the group of Len Petruccelli had shown that actually the inclusion of this particular exon was abnormal in patient tissue from FTD patients. And this was really the first evidence that indeed there was a loss of function of the nuclear TDP43 function in patients with TDP43 protein disease. But so the issue we are facing is that TDP43 regulates many targets. And so one question we have not yet answered, and our major effort on this, is to know whether some of these RNA alterations play actually a role in disease progression. And then the next phase of my talk, I will describe why we think that STAT-MIN2 is an important target of TDP43, and this is a collaboration with Don Cleveland's group and an independent work was published at the same time from Ksenia. And really the reason why initially we focused on STAT-MIN2 was very simple. We had done RNA sequencing in cells where we had depleted TDP43, and you can see that the main RNA that was downregulated was encoding STAT-MIN2. So indeed, at the protein level, when you reduce TDP43, you almost lose the formation of STAT-MIN2. And what we realized when we looked at the RNA-seq data is that in control cells, you see the reads of the RNA-seq map to the 5 exons encoding for STAT-MIN2. But when we deplete TDP43, there are reads mapping to the first intron, supporting that there is an abnormal inclusion of an exon within this first intron. And we call this exon exon 2A. And so we looked more carefully at this region, and then there were many different interesting features. So this exon contains a premature stop codon, but it does also contain TDP-43 binding sites, as well as an alternative polyadenylation site. So we looked at CLIP-seq data that were available from human cells. And indeed, you can see TDP-43 binds exactly at this region in human cells. And so what we proposed was that TDP-43 normally binds and does block or repress the inclusion of this exon, leading to the normal formation of mature RNA that encode for the full-length statin-2. But when TDP-43 is deficient, there is a splicing and inclusion of this exon 2A, and then we can detect this abnormal transcript either by RT-PCR in cells with a knockdown for TDP-43 or by RNA-seq, as I have shown you. We have also shown that there is not a downstream splicing toward exon 2, but rather the usage of this alternative polyadenylation site. And so that leads to a truncated transcript that suppress the formation of the full-length, and this Western blot where we see that full-length statin-2 is not produced when TDP-43 is deficient. An interesting observation was that actually this polyadenylation site and the TDP-43 binding site actually are not conserved in mice or rats. And that has important implication for all of the efforts to model TDP-43 proteinopathies in rodents, where we know now that the RNA target, the most affected in human cells, is actually not affected in mouse when TDP-43 is deficient. And indeed, in our initial work that we had done, knockdown TDP-43 in mouse brain, we had never identified statin-2 as a target. So we had, seen this very strong alteration of statin-2 in human cells after knocking down TDP-43. So we wanted to see what's the relevance and is it really happening in other models. And so we did several things. First of all, we introduced TDP-43 mutation in human cells. And again, this led to a downregulation of statin-2 with this abnormal exon 2A inclusion and with this deficit in the protein. We then looked at neurons. We had directly differentiated from human fibroblasts from patients with different TDP-43 mutations. And again, statin-2 is altered in these lines. We then looked at the level of statin-2 in motor neurons. And so this was very helpful to use actually some data that our collaborator John Ravitz had generated by doing RNA-seq from laser-captured spinal motor neurons. And what you can see is that actually statin-2 is in the top 25 mRNA expressed in motor neurons. So it's a neuronal gene, very important and very highly expressed in motor neurons. When we looked at RNA-seq from control versus sporadic ALS patients that do have TDP-43 proteinopathies, you can see that there is this low level of statin-2, and there is also the abnormal exon 2A. That we have actually identified in all of the sporadic ALS patients for which we had laser capture motor neurons. We also took the bulk RNA and did just the RT-PCR I have mentioned before to amplify this abnormal transcript. You can see we can detect it in sporadic ALS and not in control. We can also detect it in patients with C9orf72 disease, which are known to develop a TDP-43 proteinopathy, so it's not surprising. But what was very important is that we do not detect it in patients with SOD1 mutation. And these patients are basically the rare cases for ALS where there is no TDP-43 pathology. And so it clearly correlates to TDP-43 dysfunction. Later on, Len Petruccelli's group did a large cohort with frontotemporal dementia and showed that patients with FTLD TDP indeed have the truncated statin-2, which is not found in the other forms of FTD with tau or FUS disruption. So clearly statin-2 altered processing is consistent with the established TDP-43 pathology in ALS and FTD patients. So statin-2 is highly depleted in cells and in tissues from patients. And It was important also that it's a tubulin binding protein involved in microtubule dynamic instability. And so that raised, of course, our interest. And looking at the literature, it was actually known that loss of statin in Drosophila was associated to impaired axonal transport and neuromuscular junction degeneration and paralysis. So we are now doing several efforts to model loss of statin 2 in rodents. And one approach that we have taken in adult mice is to deplete statin 2 because in patient, this is basically what's happening. Statin 2 depletion is a downstream effect of TDP-43 dysfunction in adults. And so we wanted to model that. For that, we have done two collaborations, one in collaboration with Ionis Pharmaceuticals, where we are using antisense oligonucleotides to knock down murine statin 2 in wild-type mice using intraventricular delivery. And another one is a collaboration with Martin Marsala at UC San Diego, was recently shown a new way of injecting AAV in the spinal cord for focal depletion of targets using shRNA. And we do that with an shRNA against statin 2. So first, the ASOs were injected in adult mice, and as expected, based on a lot of work we have done previously with Ionis Pharmaceutical, when you have ASOs injected in the ventricles, it does diffuse throughout the central nervous system and knock down the gene of interest, both in the cortex and in the spinal cord at the RNA level and the protein level. And we know that these ASOs act for several weeks, and you can see that there is still a knockdown 8 weeks after the single injection. So in this mice with a low level of statin 2 using different ASOs, we have seen an alteration of the synaptic nerve conduction velocity, but no alteration of the compound muscle action potential. We have also looked at neuromuscular junction, and in mice with a control ASO, most of the neuromuscular junction are innervated, but we see a partial or full denervation of the neuromuscular junction in mice treated with ASO against statin 2. Then we used this method that has been recently published by Martin Marsala, where you do a subpial injection of AAV, and this transduces very efficiently and focally the level of the spinal cord that you target, in this case, the lumbar spinal cord. And you see that there is retrograde diffusion of the through the neurons of the AAV, and you can identify in this case, the GFP in different regions that are connected to this lumbar spinal region. So we have done that using an AAV9 encoding an shRNA for statin 2. And you can see that 4 weeks after lumbar delivery, we see a reduction of statin 2 in the lumbar region, but not in the cervical region, compared to the mice with a scrambled shRNA. When we look 32 weeks, it's clearly a sustained depletion with a clear knockdown of statin 2 in the lumbar region, with a partial diffusion to the thoracic region, but with preservation of the level of statin 2 in the cervical region. I've not told you, but statin 2 is part of a family of statin 1 to 4, and so we wanted to make sure that our AAV is targeting only one of them, and indeed the other one are preserved, as well as some targets of interest. And we did some longitudinal analysis in these mice, and during 2 months the mice are fine, but you can see that over time there is a progressive reduction of the hind limb grip strength, but the forelimb grip strength, as expected, is preserved, since we know that the cervical statin 2 levels are also not affected. We also see a time-dependent alteration of clasping in this mice. Again, as we had seen in the ASO depleted mice, we see nerve conduction velocity reduction without alteration of the CMAP. And what is very clear is that knocking down statin 2 in the spinal motor neurons is not leading to the spinal motor neuron loss. And so in the spinal cord, we see the cell bodies are intact. even after the knockdown, and we don't see macrogliosis, and we don't see astroglial increase in these mice. However, what we see again is that there is a retraction of the neuromuscular junctions with denervation of the neuromuscular junction in these mice. So this is ongoing work. These mice are being analyzed, but so far what we know is that there is a suppression of statin 2 in the adult nervous system leads to neuromuscular junction disruption, reduced nerve conduction velocity without leading to motor neuron death or glial activation. We have a very large collaboration with Catlux, who is at Jack's laboratory, and has generated a different model to test statin 2 contribution in disease, and in particular using statin 2 knockout mice, as well as transgenic human statin 2, and as well as some humanized statin 2, where we have put the human exon 2A within the mouse gene in order to recapitulate the sensitivity of statin 2 to TDP-43 dysfunction. So that's ongoing. Hopefully next time I will show you more results, but for now I'm going just to focus on our experiments in cell culture using iPS derived motor neurons from human cells, where we have knocked down TDP-43 using antisense oligonucleotides. And as expected, this led to a striking reduction of statin 2 level and the abnormal production of this truncated statin 2. Independently, we are also using statin 2A ASOs to knock down statin 2 without altering TDP-43. But in both cases, whether you knock down TDP-43 or knock down statin 2 directly, both these motor neurons have very low level expression of statin 2 protein. So we grow these motor neurons in chambers, and we treat them with ASOs for 20 days. Then we do a mechanical exotomy, and we let the neurons recover for 24 hours. And what you will see are this part where we look at axonal regeneration in the axonal compartments after 24 hours. So in iPS motor neurons treated with control ASO, the axons regrow within 24 hours, and it's very clear that statin 2 is enriched in the axons as well as in the axonal growth cones. However, in the neurons that have been treated with statin 2 ASO, after 24 hours, there is really no regeneration. So statin 2 is necessary for axonal growth. And if we have neurons that have a statin ASO, but then are transduced with an antivirus expressing statin 2, we can correct this phenotype. In motor neurons with TDP-43 ASOs, I showed you that the level of statin 2 is very low. So not surprisingly, there is no regeneration of the axons. But where we were surprised is that in these motor neurons that have TDP-43 depletion and hundreds of RNA alterations, if we restore only the statin 2 level, this phenotype is completely rescued. And so restoring statin 2 is sufficient for regeneration despite hundreds of RNA alterations that are dependent on TDP-43 depletion. And so that was very encouraging and led us to propose that we should find ways of restoring statin 2 in TDP-43 deficient cells. And of course, there are several strategies. One that we are taking in collaboration with IMS Pharmaceutical is to use antisense oligonucleotides. And so ASOs can act different ways. One way of the ASOs, depending on the chemical modification, they can block splicing of target genes. And this is the approach that has been developed to block the abnormal splicing of SMN in another motor neuron disease called spinal muscular atrophy, and this is an FDA-approved drug. So we took a similar approach for statin 2, and we have used the cells that I've shown you have mutation in TDP-43. They have low level of statin 2, a full length, and they have this abnormal truncated statin 2. And so for that, we have looked at these cells treated with different ASOs, and we have identified ASOs that are able in a dose-dependent manner to restore the level of full-length statin 2. And they do that by blocking basically the abnormal splicing and the abnormal formation of the truncated RNA. So we have used these ASOs now in iPSC-derived motor neurons, really using the same paradigm that I mentioned, we treat the iPS motor neurons with ASOs against TDP-43 to deplete TDP-43. Then we treat them with these rescue ASOs that are blocking the abnormal splicing of statin-2, and we look at regeneration after axotomy. So in these iPS motor neurons, again, in a dose-dependent manner, different ASOs are able to increase the level of statin-2, to decrease the level of the abnormal truncated mRNA. And as I've shown you before, the motor neurons, when they're treated with TDP-43 ASOs, they do not regenerate. But if we block the abnormal splicing of statin-2, we are able to, again, rescue this abnormal phenotype and have axonal growth being restored. So these ASOs are under testing now in vivo in mouse models, including with the collaboration with Clotilde that I have mentioned. But as I said, we have also, I think, other ways of modulating statin-2 levels. And Matthew Nolan in my group has made several cellular models in order to perform a small molecule screen. He has now identified some molecules that do regulate the level of statin-2, and we will be able to confirm these molecules in the different models we have. We have also generated some cellular models with the goal of doing a CRISPR screen with collaborators to identify modulators of statin-2. So to conclude this part, I've shown you that alteration of statin-2 is a hallmark in patients with sporadic ALS and FTD. And we are now looking at the implication of statin-2 in other neurodegenerative diseases. And of course, in Alzheimer's disease patients, we know that 30% of them have the TDP-43 proteinopathy. So we're looking at that. I've shown you that reduction of statin-2 in adult mice induces progressive motor deficits and axonal withdrawal from the neuromuscular junction, and that we can restore statin-2 level in iPSC-derived motor neurons, and that's sufficient to rescue axonal regeneration in TDP-43 deficient motor neurons. And we are testing basically if this approach could be a new therapeutic strategy in ALS and FTD. So now do we have time? Yes, I think. So for the third part, I'm going to focus on C9orf72, and that is a very, very interesting and very specific form of ALS and FTD. It was found in 2011, and as you can see on this graph, it's clearly the most frequent cause of genetic cause for these conditions. But what's really particular is the type of mutation that is involved here. And so this is basically a repeat, an exonucleotide repeat, G4C2, that is normally repeated 2 to 30 times in the population. But in patients, there's an increase of the number of repeats to hundreds, if not thousands of repeats. Another particularity is that the repeat lies in the non-coding region of the C9orf72 gene. And because there are different start sites that can be used, you can see that it does lie either in the first intron or in a promoter region, depending on which isoform of the C9orf72 transcript we are looking at. So because it's a repeat expansion, I think the field has really early on benefited of the 20 years of work that had been done in other repeat expansion. And very quickly, several disease mechanisms were proposed that are reminiscent to what was known in other neurodegenerative disease with repeats, but also that are supported by observation in tissues and cells from patients. And so very schematically, two types of mechanisms were proposed. One is loss of C9orf72 function, and that is supported by indeed, there is a reduction of the level of C9 of RNA in presence of the repeats, and it's been shown that there are epigenetic changes and repression of the transcription of the downstream isoforms in presence of the repeat. Another mechanisms is a gain of toxic function that can be coming from different species that are generated by the repeat. And one is that the expanded transcript do accumulate into RNA foci. And this is something that had been previously shown in myotonic dystrophy. And in myotonic dystrophy, it was clearly demonstrated that these RNA foci sequester RNA binding proteins called muscle blanc, leading to RNA processing alterations. It's been proposed that similar mechanisms exist in C9orf72 disease. There are clearly RNA foci. I have to say that it's still very controversial which, if any, of the RNA binding proteins are indeed sequestered in the RNA foci, and many of them have been proposed. Another gain of toxic function is a mechanism that is relatively recently described by Laura Renan. And she first described that in myotonic dystrophy and SCAR8. And this is the fact that an RNA that contains an expansion can be abnormally translated, actually in all frames, even if you don't have a translation codon. And that leads to aberrant proteins from regions that are not in the coding region. And these proteins coming from the repeat accumulating in patient tissues. And indeed, in C9orf72, several groups, including the one from Laura Renan, has shown that the C9orf72 repeat is abnormally translated, leading to accumulation of proteins, different types that we call dipeptide repeat proteins. So these different disease mechanisms are proposed. They are supported by some observation in tissues from patients, and they are not mutually exclusive. But it was very important for the field to dissect, what is the contribution of each of them. And so an approach we took was to model these different mechanisms using mice. So the first experiment that I did when I was still in San Diego was to knock down C9orf72 using antisense oligonucleotides again in wild-type mice. And then we looked at really the genetic model, so C9orf72 knockout mice, and we generated also back transgenic mice expressing the repeat to test the gain of function. More recently, we have tested if a combination of loss and gain contributed to disease by crossing these different models. So I'm going to glance through these different models relatively quickly. The loss of function using ASOs really was extremely well tolerated. We didn't observe any phenotype. However, the knockout mice, we had generated heterozygous and homozygous mice. What you can see is that, first of all, loss of C9, even homozygous loss of C9 is not essential for development. The mice are fine, and they are fine for many months. The heterozygous mice do not develop phenotype. However, the homozygous started to die. And what we realized is that they developed a peripheral phenotype with this large splenomegaly and large lymph nodes. And so this was actually completely independently shown by many groups who have used independent C9orf72 mice, and clearly showing that C9orf72 protein is very important for immune response. And there is some very interesting ongoing work to see what's the contribution of C9 in the immune response and maybe its contribution to disease. But clearly, none of us have really observed neurodegeneration or any pathological features reminiscent of ALS or FTD in this mice. So the loss of function was not enough to recapitulate disease. As I said, in parallel, we had generated back transgenic mice. We used a human back that does express 450 repeats. There's a few specificity. First of all, the C9 gene is truncated, so we do not overexpress full-length C9, which we thought was a good thing. We don't know if it's toxic to overexpress. And there is no other genes in this back. We have made different lines. Actually, funnily enough, it was DB Swing, who Joanna has just mentioned, who also injected these mice. So I think we have the same good ends. And she generated many, many transgenic lines that we characterize. I'm not going to go through that because we compared the lines based on different size of the repeat, different expression level. I'm just going to focus on one of these lines that express 450 repeats and clearly does recapitulate some of the pathological features of C9. First of all, these mice do accumulate RNA foci, both from the sense strand and the antisense strand, which is exactly what is seen in the patients. And importantly, they also accumulate dipeptide repeat proteins. So I have mentioned this phenomenon by which the expanded repeat is being translated in all frames, actually both from the sense strand and the antisense strand. And several groups have shown that indeed this dipeptide repeat protein accumulate in patients. And there is a wealth of studies now showing that these dipeptide repeat protein have a toxic effect, some more than others. And so these transgenic mice accumulate the dipeptide repeat proteins. However, they absolutely do not recapitulate an ALS model. We have not seen motor neuron loss. We have not seen motor deficits. The spinal motor neurons are conserved. What we have observed is a deficit in spatial learning that was using bounce maze. You can see over the months, the transgenic mice do perform less well than the wild-type mice. We have seen that actually in two different transgenic lines, where the mice find this black box less easily than the wild-type mice. This deficit in spatial learning was confirmed using a radial maze. Again, the two transgenic mice are deficient in this task. There is also apparently an increased anxiety that is seen by marble burying and elevated plus maze in these mice. These cognitive deficits are consistent with the loss, very mild actually, but reproducible between lines of hippocampal neurons. So this is a very mild model, but considering that there was this pathological accumulation of RNA foci, dipeptide repeat protein, with the behavioral alterations, we decided to use this mouse model with Ionis Pharmaceutical to develop C9orf72A cells. This time we use this type of mechanism that engage in RNase H and degrades the target RNA, in this case C9orf72. And so we have tested ASOs first in cell, but I'm just going to show you the results in mice, where a single injection in the transgenic mice was enough to reduce the accumulation of foci, was enough to reduce the accumulation of dipeptide repeat proteins. It was, again, in this completely independent cohort, where we saw again the deficit, in the cognitive deficit. This was rescued by the antisense oligonucleotides. The marble burying and elevated plus maze were also improved in mice that had received ASOs, reducing the level of C9orf72 with the expansion. So based on these preclinical results, Biogen has started a program to develop ASOs, and there is now a clinical trial in C9orf72 ALS patients testing these antisense oligonucleotides to target C9orf72. So we think it's a very promising approach, but we still continue to actually investigate alternative methods to target C9orf72. And in particular, we are very interested in what are the mechanisms of the formation of dipeptide repeat protein, and could we target this directly. And we and others have investigated what are the components that influence the translation of the C9orf72 repeat. And in particular, Ricardo Tabets in my group, in collaboration with Frank Martin in the University of Strasbourg in France, have recently shown that the C9orf72 repeat is actually preceded by a CUG codon that can be used as a translation initiation start site. So the ribosome use that to translate the poly-GA, which is in the same frame. But interestingly, what we realized is that it was actually also necessary for the translation of the two other frames, GP and GR. And I'm showing you just one slide of this published work where when we mutated the CUG that precedes the repeats into a CCG, both in cell-free system and in neuronal cell cultures, the mutation of the CUG into CCG alleviates the formation of the dipeptide repeat in the three frames. That is strongly supporting that the ribosome starts here, but then there's a frame shifting, and that it's then going to go to both frames to produce poly-GP and poly-GR. But importantly, when we mutate this CUG into CCG, it has no impact on the formation of RNA foci. So we still have the transcript with the repeats that do accumulate. And so this really gives us a great opportunity to test what is the relative contribution between the RNA foci toxicity and the dipeptide lipid protein toxicity. And so in collaboration with Len Petruccelli, what initially showed that you can use AAV injection of G4C2 repeat at P1 in animals to have a mouse model that actually does recapitulate many of the C9orf72 features. We have done the same, but using constructs that either contain CUG or contain CCG. And Len Petruccelli had shown mice with either 66 repeat or 149. So we have also both models that we are testing. And so Xin Zhang in my group, who is leading this project, has injected and characterized now. She's in process of characterizing these mice at different age for different behavioral and pathological analysis, including in collaboration with Ksenia and Elisa, the neuronal activity in vivo. And so this is ongoing work. I'm just showing you preliminary results that clearly show that even in vivo, when we mutate just one base of this CUG to CCG, we prevent the formation of dipeptide lipid protein, despite the formation of RNA foci. You can see in all the three frames, we don't have accumulation of the dipeptide lipid protein. But very interestingly, preventing the formation of the dipeptide lipid protein basically alleviated the behavioral phenotype of these mice. So these mice had been shown with a CUG66 repeat to have abnormalities in open field, marble burrowing, inverted grid, hanging wire. In all of these tests that we looked at, the mice in blue that have this CCG mutation do not have dipeptide lipid protein alteration and do not have the behavioral phenotype. And there is also this interesting marker, neurofilament, that, as you may know, is developed as a biomarker for ALS, but also for other neurodegenerative disease where NFL is increased when you have axonal and neuronal loss in neurodegeneration. And in these mice, indeed, we can show that NFL in the plasma is increased, but that is corrected in the mice with the CCG. So this is ongoing work, but I think it strongly suggests that the dipeptide lipid protein in these mice are really triggering the phenotypes that are observed, rather than the accumulation of RNA foci and putative RNA binding protein sequestration. So I'm going to finish with this conclusion slide where I've shown you that complete loss of C9orf72 leads to immune dysfunction with peripheral splenomegaly and large lymph nodes. That the mice modeling gain of function mechanisms recapitulate behavioral and pathological features of disease, and that ASOs targeting specific C9orf72 isoform containing G4C2 represent a promising therapeutic approach in C9 ALS FTD. And more recently, we have evidence that production of the dipeptide lipid proteins from all frames can be alleviated by mutating the CUG initiation codon, and that leads to the rescue of the C9orf72 disease in vivo. So what I've presented is highly collaborative, and I'm sure you have seen it already over it. So I want to thank the members of my lab. I didn't have space here to have all the alumni, but several of them have been involved. I have a very close collaboration with Don Cleveland's group, in particular on STAT2, and actually on C9 also, and with Ionis Pharmaceutical for different approaches. I've mentioned Martin and Mariana, who has done the subpial injection. I've not shown new results with catlets, but we have this ongoing characterization of several mouse models and a very long standing collaboration with Len Petruccelli, as well as many of you here. And I thank you for your attention.